This begins the second part of our muscle physiology lecture. We're going to continue and pick up with muscle metabolism. So for muscle metabolism, one of the first questions we have to ask is, how do muscles get the ATP or energy necessary to power the contraction cycle that we covered in the last tutorial and the last PowerPoint? Well, the answer is one of three ways. So the first is going to be creatine phosphate. Now, for each of these, we're going to outline how many ATP are produced and how much work is going to result. The second is anaerobic glycolysis. The third is going to be aerobic respiration or cellular respiration. Let's start with creatine phosphate. Now, creatine kinase catalyzes the transfer of a phosphate group from creatine phosphate to ADP, which rapidly yields ATP. So again, we utilize an ATP, we hydrolyze it to get that phosphorus group, we get creatine to creatine phosphate, which gives us energy for the contracting muscle. Now, what does this give us? It gives us one phosphate. That one phosphate is going to provide a relatively short contraction duration, roughly 15 seconds. So we're going to rank these as to how much time or energy we get. So with creatine phosphate, we get one phosphate, which is going to give us about 15 minutes worth of energy for work. The next process is anaerobic respiration or anaerobic glycolysis. So this is going to be our second tier. When creatine phosphate stores are depleted, glucose is going to be converted into pyruvic acid to generate the ATP necessary. So let's look at our start. We start with muscle glycogen. We're getting glucose from the blood. We're undergoing that process of glycolysis, which is going to yield 2 ATP. So if you remember, with creatine phosphate, we got 1 ATP. With anaerobic glycolysis, we're getting 2 ATP. So we're getting a little bit better with the amount of energy we're getting to sustain work. Now, the byproduct of this process is going to be 2 pyruvic acids that's going to be converted into 2 lactic acids, which will be pushed into the blood to be removed. Now, if you've ever had muscle cramps or muscle pains after exercising a lot, it's due to using these glucose stores, the glucose and the glycogen stores in the muscle, and the metabolic byproduct is lactic acid. So until you clear that lactic acid, you get those muscle pains. Now look at our time. We're getting 2 minutes of work here, 2 minutes of energy to contract. So before we go 15 seconds with creatine phosphate, now we're getting better with 2 ATPs to give us about 2 minutes worth of energy for contraction. Our last process is aerobic respiration. This is cellular respiration. So, under aerobic conditions, pyruvic acid can enter the mitochondria and undergo a series of oxygen-requiring reactions to generate large amounts of ATP. So, we go and going to sidestep for a little bit and talk about a hormone, the hormone being T3 and T4, which is a BMR, basal metabolic rate hormone from the thyroid, and its job is to monitor oxygen consumption by cells. So everybody doesn't really understand, what does that mean, oxygen consumption by cells? Well, here it is. These cells are taking that oxygen and running these reactions, which are going to generate large amounts of ATP. So let's look at the process. So fatty acids are going to be liberated from adipose cells. This is called freeing fatty acids. We're going to see amino acids from protein breakdown, pyruvic acid from glycolysis, and oxygen from hemoglobin in blood or from myoglobin in muscle fibers is going to run the Krebs cycle and electron transport chain in the mitochondria. Now, what's the byproduct? We're going to get heat, we're going to get carbon dioxide, cellular respiration, and water, and more importantly, 30 to 32 ATP. So we exponentially increase the amount of ATP. So now you can guess the duration of work is going to be several minutes to hours worth of energy. So if you start thinking about muscle and which muscles are contracting for a longer period of time. Think about your leg muscles and how they are resistant to fatigue, or all the muscles that keep your spine erect or your head up. They're resistant to fatigue because they're relying on cellular respiration to produce this massive amount of ATP so that they can contract for minutes or even hours throughout the entire day. Energy sources and fatigue, we do have an interaction animation, this muscle metabolism, so it's the role of ATP and muscle movement and fatigue. Now, you can click on this in your PowerPoint slide, not on this tutorial, but you must be connected to the internet in order to run this animation and in slideshow mode in order to see this little video. The next topic we're going to cover is muscle fatigue. So we kind of introduced this word in the previous slide when we talked about using energy and being resistant to fatigue. So what is actual fatigue? So we'll define it. Muscle fatigue is the inability to maintain force of contraction after prolonged activity. Now, what causes the fatigue? So again, go back to neuromuscular junction and sliding filament theory. Remember those rate limiting steps? Well, here we go. So an inadequate release of calcium, so not enough calcium from the sarcoplasmic reticulum. Remember, calcium equals contraction. No calcium, can't contract. No acetylcholine, can't contract, right? So those rate limiting steps. Now, let's also deplete our energy stores. We deplete creatine phosphate. There goes that short burst of energy. We deplete oxygen. There goes aerobic respiration. And nutrients, that's fueling all these processes. We build up lactic acid and ADP from our anaerobic processes. Okay, so this can also fatigue our muscle. And then lastly, going back to sliding filament theory, those rate limiting steps, insufficient release of acetylcholine at the neuromuscular junction. So remember, no acetylcholine, no depolarization, no release of calcium. Okay, so that leads to muscle fatigue. So the question now is, well, once you did all this exercise and now your muscles are fatigued, how do you repay it? So we talk about oxygen consumption after exercise. Now what happens? We rely on the cardiovascular system and the respiratory system, which will allow you to breathe heavily for a period of time after stopping exercise. We're doing this to pay back our oxygen debt. Now where does this extra oxygen go to? Well, it helps to replenish those creatine phosphate stores. We're also converting lactate into pyruvate, and we're reloading oxygen onto our myoglobin. So just again, a little digression. If you heard about after exercise or during exercise, people drinking creatine, this is helping to replenish those creatine phosphate stores. So it is a way that we can pay back those lost little energy stores. Control of muscle tension. So our next topic is going to be to discuss how we control muscle tension. So the strength of a muscle contraction depends on a couple things. How many motor units are being activated, the type of motor unit, so are they small or large, okay? So let's define what a motor unit is. A motor unit consists of one somatic motor neuron and the muscle fiber it innervates. Activating only a few motor units will generally result in a weak muscle contraction. Activating many motor units will generally result in a strong muscle contraction. So I usually use an analogy here thinking of an army, and this also helps to introduce a new term, which is recruitment. So if you are going to fight a war and your army is relatively small, you're not going to do very well or succeed against fighting a huge country. So how would you strengthen your army, strengthen your force? The answer is recruitment. You recruit more soldiers to make your force bigger, ultimately stronger. Muscles do the same thing. So weak contractions recruit fewer motor units, whereas stronger contractions are going to recruit many motor units. So we're going to define motor unit recruitment. It is the process in which the number of active motor units increase. There is a process. Weak motor units are recruited first, followed by stronger motor units. And ultimately, motor units contract alternately to sustain contractions for a longer period of time. The next part of the contraction is to look at a single cycle of that contraction. A single cycle of that contraction and relaxation period produced by a stimulus is called a twitch. As you increase the frequency of stimulation, a muscle will develop more tension and undergo sustained contraction that reflect normal muscular activities. Now, there are three phases of a twitch. There is a latent phase, a contraction phase, and a relaxation phase. So the names kind of help us explain what they do. In the latent phase, this is where we're kind of waiting. So acetylcholine is released into the synaptic cleft, so go back to that neuromuscular junction. We're depolarizing the sarcolemma. Calcium is released from the sarcoplasmic reticulum. And this is going to lead to, so remember, calcium equals contraction equals our contraction phase. What's happening? Sliding filament theory. Cross-bridge cycling occurs. The sarcomeres shorten and muscles contract. Now, remember at the end of that sliding filament theory, we undo it. So we recall calcium back to the sarcoplasmic reticulum with those ATPase gated recall channels. This is our relaxation phase. Actin binding sites are covered and muscle resumes its relaxed length. Now we could put it to picture, put it to graph, which is going to be in our following slides. So let's look at the stages, those three stages of a twitch contraction. So the brief contraction of all muscle fibers in a motor unit in response to a single potential. We start with this latent period, right? That's when that ACH, the acetylcholine, is moving into the cleft. We're depolarizing the sarcolemma, which is allowing for calcium to be released, which leads us into this contraction period, right? So muscle contracting, we go sliding filament theory, cross-bridging, and then we detach with ATP, and we enter into our relaxation phase. And then we hit a refractory period, which is waiting for another stimulus to come to allow for the cycle to recur again. Now, as those action potentials come, it is the frequency of the stimulation that is going to give us stronger contraction. So, we look at different types of stimulations. The first is wave summation. This is when a second action potential triggers muscle contraction before the first contraction has finished. What this does is it allows us to build up on strength, so we result in a stronger contraction. Now we break apart two types of tetanus, and you probably heard this word before. There's a bacteria that causes lockjaw. It is called tetanus. You get shots for this every 7 to 10 years. So what we're trying to do is prevent that bacteria from causing a complete or incomplete type of tetanus. That's that lockjaw scenario. So there's two types of tetanus, fused and unfused. It's also called incomplete and complete. So we're going to put it to graph and identify an increase in stimulation versus the muscle tension. The first one we're going to look at is a trepa. This is a repeated stimulation immediately after relaxation phase has been completed. Note that an increase in tension observed compared to the previous contraction. This is producing a staircase effect because with each stimulation, muscle tension increases. Now, what causes this? Well, it's due to an increase in calcium from through the sarcoplasm that wasn't taken back to the terminal cisterns or the sarcoplasmic reticulum. This is essentially a warm-up. So when you go to the gym, you go on that treadmill and you warm up your muscles, you are undergoing this type of increase in stimulation, a trepa. Now we could change and affect the type of stimulation coming in, make it come closer together, which will change the contraction. But on the next slide, we're going to look at what the trepa looks like. So this gives us that little staircase, that build up, that warm up effect. As time is increasing, we are increasing tension. But notice our cycle, our latent period, our contraction, and our relaxation period is almost allowing it to fully come to an end, which builds up this nice little staircase. Now, if you're guessing, well, how do we increase tension? Somehow we have to go this way. So how do we do that? Well, let's look at our next scenarios and we'll see how we build up this tension. Number 2 is wave summation. So now here's that scenario. We need to get more tension. The second stimulus arrives before relaxation completes. So on that come down on that graph, before it is allowed to go to baseline, we hit another stimulus. Note that the intensity of the stimulus is always the same. We cannot change the stimulus. A stimulus is a stimulus. You cannot make the mass of the stimulus greater. All you can do is increase the frequency between the stimuli. Wave summation, one twitch is added to another. The increase in tension results from the muscle already being partially contracted from the previous stimuli. So let's look at the graph on the next slide. Now what we see here is this increase, massive increase in tension. How? Well, we latent, we contract, we relax, and before we're allowed to relax away, boom, another stimulus comes. We run that cycle again, boom, another stimulus comes. We run it again before we're allowed to relax, boom, another stimulus comes. What this does is it allows us over time to build an increase in tension. So we're building more strength before that stimulus ceases and we come back to relaxation. The next two that we're going to finish with is incomplete and complete tetanus. So let's start with incomplete tetanus. This is where the muscle never relaxes completely. So as you can envision, we're cutting off that relaxation period really soon. So stimulus is coming really rapidly and quickly, preventing that relaxation, which allows us to build tension relatively fast. So here the stimulation is rapid, action potentials arrive so frequently that only some relaxation is possible. So let's look at the slide on the next page here to work through what happens. So look, here we go. We're increasing tension, not to maximum tension that we see in complete tetanus, but we're getting pretty close. So we relax, latent, contract, relax, boom, another stimulus. Latent, relax, boom, another stimulus. Latent, contract, relax, boom, another stimulant. Contract, relax, boom. And what this happens is we get to a point where we plateau out, where we're getting a significant amount of tension to do some strong work like lifting a rock or holding that book straight out in front of you off the table for a significant period of time. The last one we're going to talk about is complete tetanus. This also happens to be one of the most painful ones. If you've ever had a Charlie horse or leg cramp, you understand what complete tetanus feels like. This is where relaxation phase is completely eliminated. So action potentials are coming constantly. Calcium is never taken back to the terminal cistern or the sarcoplasmic reticulum. Maximum tension is reached and sustains a permanent contraction. Look at the graph. We go from latent to contraction, no relaxation phase. We hit all the way up to maximum tension. If we were to go any higher than this, the muscle or tendon would rupture and would rip. So this can be excruciatingly painful. If you ever heard of rigor, which is a symptom of diseases like malaria falciparum, one of the latter stages before you die, your body enters into this rigor, so this complete tetanus, a complete contraction, is excruciatingly painful. So we can look at a myogram, so a gram that overlaps muscle contraction with force of contraction with action potential. So we put together our 4 different types of contraction with the frequency of stimulation. So notice the difference in contraction time and force produced for each of the conditions below. So again, we start with a single trepa or twitch, right? And then you see that our force is relatively low. We get a full cycle. 1 action potential. Then we have wave summation. So again, summation, we're summating, we're adding these peaks. And again, you notice we come up and then another stimulus, so first stimulus and then a second stimulus comes before we hit relaxation and we throw another peak in. Now, unfused tetanus. Again, more stimuli coming, not allowing the muscle to relax. 1, then the second one strikes, then the third, and the fourth, giving an increase in tension. And this is incomplete tetanus. And then with complete or fused tetanus, look how quickly the stimuli come, right? And it's giving this full-blown contraction, a real fast incline all the way to the top of a max force of contraction until the stimulus ends and then the contraction ceases. The next concept we're going to discuss is muscle tone. Now, even when at rest, skeletal muscle exhibits a small amount of tension. This is called tone. Tone is established by the alternating involuntary activation of small groups of motor units in a muscle. So when you look at an actor or actress that is really toned, their muscles are pushing through their skin. This is those small little twitches that are occurring throughout the day that allows for this high level of tone. that brings us to our various types of contractions. So we introduced some of these words earlier in this lecture, but now we're going to elaborate. We have isotonic contraction versus an isometric contraction. Now, with isotonic contractions, we have two varieties. We have a concentric and eccentric. So let's define isotonic. This is tension is constant while muscle length changes. So with a concentric contraction, this is a classic contraction where the muscle physically gets shorter and fatter at its belly. An eccentric contraction is the opposite. It typically opposes concentric contractions. So this is where the muscle elongates, so it gets longer with the contraction. So if you think about the bicep and tricep as antagonistic muscle groups, the bicep, when you flex the forearm, bring your wrist or hand to your shoulder, this is flexion of the forearm. Your bicep is undergoing a concentric contraction. Whereas the tricep, the antagonist here, is elongating, opposing that force, producing strength, which is an eccentric contraction. Then when you flip it and you extend your forearm, the muscle groups switch. The bicep brachii becomes the eccentric muscle, and the tricep becomes the concentric muscle. Now the next is isometric. Now an isometric muscle contracts but does not change length. And the example would be if you were to push on the wall, you're not moving the wall, your arm's not moving, and the muscle is not changing shape, it's not getting shorter or longer, but it is producing force. Another example would be to hold a book or your laptop straight out in front of you for as long as you can. You can see a couple phenomena going on here. One is the isometric contraction of the bicep as holding that book up, opposing the force of gravity, pulling it down, and then eventually fatigue as your muscle weakens and you start losing the ability to maintain that outward straight force. So let's put it to words. So an isotonic contraction, tension is constant while the muscle length changes. So again, a concentric, tension rises above the resistance, length of the muscle shortens, produces movement. Here's our eccentric. Peak tension developed is less than the load of the muscle, and the muscle elongates. Again, this could be due to the contraction of another muscle like the bicep and tricep that we used before. Our examples of alternating concentric and eccentric contractions are flexion and extensions of the forearm at the elbow, so like when you lift weights. We went through this one already. So during flexion, the muscle shortens and overcomes the resistance. During extension, the tension produced is not sufficient to overcome the force of gravity, but it controls the speed of the movement. The eccentric contraction allows for precise control. Isometric contraction. Tension rises and the length of the muscle fibers shorten, but the length of the entire muscle remains the same, remains constant. This is because resistance is too great. Examples would be holding your head up, holding something heavy like a book or rock straight out in front of you with your arm. Muscles that are involved in sitting or standing that oppose the force of gravity are examples of isometric contraction. Here's some pictures to help you explain. The first one is flexion of the forearm at the elbow. So this is a concentric contraction right here at the bicep. And then eccentric contraction. If you let the book go down, the bicep now is elongating, opposing the force of gravity, and so this would be eccentric contraction. Isometric, no movement. That person is holding that book straight out. The muscle is producing force to resist gravity and keep it out there until fatigue occurs. The next step in muscle physiology is to look at skeletal muscle fiber types. We have 3 different types of fibers: white, pink, and red. Our red ones are going to be called slow oxidative fibers or SO fibers. Our fast glycolytic fibers are going to be white. These are FG fibers. And then we have our FOG, fast oxidative glycolytic fibers, are going to be pink. Now, the color is going to give us some idea of how they're getting energy and doing work. So the red would tell us that there's a lot of blood coming to here. The pink, maybe an intermediate amount of blood, and the white would be very little blood. So we're going to look at a table that kind of explains everything out and puts everything together for us. So let's work through table 10.4 to understand the characteristics of the 3 types of skeletal muscle fibers. So we're going to look at a couple characteristics: structural characteristics and functional characteristics. Let's start with our SO fibers, or slow oxidative. Remember, these are our red fibers. So again, because they're red, they're going to have a lot of myoglobin, which means we're getting a lot of blood here, many mitochondria, many capillaries because of the high blood, and the color will be red. Its functional characteristics. So, because there is a lot of mitochondria, the capacity for generating ATP is going to be high, and the process is going to be cellular respiration or aerobic respiration. Rate of ATP hydrolysis. So the speed at which we use that ATP is slow. Now this is super important. Now if we're producing a lot of energy and we use it slow, the thought process is the ability to resist fatigue is going to be high because it can sustain a contraction a lot longer. So the contraction velocity is going to be slow. The fatigue resistance, look at that, high. Creatine kinase. This is the lowest amount of the three fibers. We don't need that one little ATP here. We don't need a lot of glycogen stores here. See, glycogen's low as well. Because we make so much ATP on our own, we're going to rely on that for our sole source of energy. Now, order of recruitment. First, these are going to be the first guys to be recruited because they can sustain a longer contraction. Location of where we find these in abundance. Now remember, these are scattered throughout the body, but where in abundance, postural muscles like in the back and the neck will keep us upright. Primary functions of these fibers: maintaining posture and aerobic endurance activities like long distance marathon runners. Now we go to FOG, or fast oxidative glycolytic. These are pink fibers. Myoglobin a lot, mitochondria a lot, capillaries a lot. Color red pink. Now let's look at its functional characteristics. Capacity for generating ATP. It uses both. It's intermediate. It's the middle of the road one here. It uses both aerobic and anaerobic glycolysis. Rate of hydrolysis. Now this is where we change. This is fast. So it's using up that ATP quickly, which means its contraction velocity is going to be fast. These are fast movements. Fatigue resistance. It's the middle of the road since we're producing a lot of ATP from aerobic respiration, and we're also using glycolysis to get those two ATPs. This is going to be a middle of the road and intermediate resistant to fatigue. Creatine kinase. We are going to use some here. It's an intermediate amount because when all those other aerobic ATPs run out and glycolysis ATPs run out, we're going to rely on that one creatine phosphate to give us a little bit of energy when in demand. Glycogen stores, intermediate, and that makes sense because we are undergoing anaerobic glycolysis. Order of recruitment, second. Where are we going to find these? Arms, shoulders, legs, lower limbs, right? We're going to find this for walking, sprinting, so when we're moving at a relatively slow pace, or we need a quick burst of energy, so our sprinters here. Our fast glycolytic fibers are FG fibers. These are our white ones. These guys are going to have the lowest amount of myoglobin because they're white. That's why they're not red. Few mitochondria, few capillaries give us this white pale appearance. Now, look at the capacity for generating ATP. It doesn't have a lot of mitochondria, so it's low by anaerobic glycolysis. The rate of hydrolysis for getting that energy is fast. Contraction velocity, fast. Fatigue resistance. This is the lowest fatigue resistance. Look at your arms. Think about walking on your arms. You can't do it very long. Maintaining a handstand is very hard. You fatigue very quickly. Whereas on our legs, if we compare these to our FOG fibers, our legs are able to walk for hours if we need to. Creatine kinase, highest amount. Because these guys are rapid, intense movements of short duration, we're going to rely on that creatine kinase to give us a lot of energy. Glycogen stores, yes, high. Order of recruitment, third. We're recruiting these guys last. Look at these for extraocular muscles, so the muscles of the eye that give us quick jutting movements up, down, left, right, down to the side. This is where we're typically going to find these muscles in abundance. Primary function of these fibers: rapid, intense movements and for short durations. So let's put it to real life. Let's think about it. Go back to that table and think about what type of fibers does a marathoner most heavily rely on. So again, we're talking about those slow oxidative fibers that are going to give us a long contraction. How about a shot putter? What are they going to rely mostly on? So arms, quick, rapid burst of energy. We're talking about our fast glycolytic fibers. So those are the third order of recruitment. What fibers does a soccer player most heavily rely on? So these are going to be those intermediary ones, our fast oxidative glycolytic fibers, or FOG fibers. And then what about stretching, right? If we're stretching, what are we doing? And what about strength training? So building up strength, where we use all three fibers depending on what type of strength training we're doing and what muscle groups we're working on. Now that we have finished with skeletal muscle, it's time to revisit and elaborate on cardiac and smooth muscle. Let's start with cardiac muscle. Cardiac muscle is very similar and has the same arrangement as skeletal muscle, but it also has a secondary feature called intercalated discs. These intercalated discs contain desmosomes and gap junctions that allow muscle action potentials to spread from one muscle fiber to another. Cardiac muscle cells have more mitochondria and their contraction lasts 10 to 15 times longer than skeletal muscle. So, if we had to put it to drawing, remember that peak that we saw that went up and down for a single twitch for skeletal muscle? Cardiac muscle is going to look a little different. It's going to come up and there's going to be a plateau phase. This is going to allow for that muscle to sustain that contraction longer, allowing for the movement of blood. Smooth muscle, on the other hand, is very different from cardiac and skeletal muscle. It looks completely different. It lacks striations, it's thick in the middle, and tapered on the ends. It can be arranged as either single unit or multi-unit fibers. Smooth muscle contractions start more slowly and last longer than skeletal and cardiac muscle contractions. The smooth muscle can shorten and stretch to a greater extent than skeletal and cardiac muscle, and the smooth muscle fiber shortens in response to stretch. So if you think of it as a fishing net that you would cast into the water, you pull on it, it cinches down and catches the fish. This is similar to the contraction cycle of smooth muscle. Now, if you think about the differences between smooth and skeletal, you think about that fable, the tortoise and the hare. I know we've introduced this before, but let's revisit it. So this smooth muscle is essentially slow and steady, is going to win the race. We'll find this in the digestive tract. Whereas skeletal needs to be fast and deliberate, like the hare. So table 10.5, we're going to go through the major features of the three different types of muscles and where you find them. You can go through the table and work through all the information. We've covered this throughout this lecture, so it should be a repeat, but it's a nice way to put it all together into one nice little neat bundle. So we're going to end our lecture with a few more facts about muscles. Regeneration of muscle tissue is where we're going to start. So mature skeletal muscle fibers cannot undergo mitosis. We do see a few conditions, the first being hypertrophy, which is defined as an increase and growth of muscle cells. The second is hyperplasia. This is where a single muscle cell is divided or ripped into two new muscle fibers. Now, basically what this does is it doubles the muscle mass in the individual, which causes a permanent growth in muscle that will be there for perpetuity, forever. Whereas in hypertrophy, if you stop weightlifting, then the muscle cells shrink and muscle mass decreases. Whereas in hyperplasia, that muscle mass does not shrink. It is essentially there forever because you created two new muscle fibers from the one. Lastly, we have smooth muscles and pericytes. Now, pericytes are spatially isolated cells that typically surround capillaries. They work together with vascular smooth muscle cells that surround large blood vessels like arteries, arterioles, venules, and veins. They make up the cells that are going to support blood vessels. Now, as we wrap up this lecture, the last thing we're going to look at is what happens to our muscles as we age. So, ultimately, aging and muscle tissue. Now, this is an interesting fact. Between 30 and 50 years of age, about 10% of our muscle tissue is replaced by fibrous connective tissue and adipose tissue. Between 50 and 80, another 40% of our muscles are replaced with connective tissue and adipose, which is why the elderly have such a difficult time with flexibility and movement. So here's our consequences of this. Muscle strength and flexibility decreases, reflexes slow because of less muscle, and slow oxidative fiber numbers increase to keep us upright.